Rennie and colleagues. So let me try to put my slides in the meantime. I want to thank for this invitation. Are you looking at my slides?
It's clear.
Hello. Can you see my slides?
Yes.
Yes.
Okay. Okay. So I would like to describe very quickly what is going on with CoronaVac in Brazil since we started the collaboration with Sinovac. So I will have 3 points to discuss. One is the pivotal phase 3 trial that we have the acronym PROFISCOPE. The other one is the Proyecto S, that is an effectiveness study that is ongoing, and lately to discuss a little bit about our current immunization program in the country. So first of all, I want to introduce you Butantan Institute, for those that are unaware of. This is a public health research institution linked to the Secretary of Health of the state of Sao Paulo. So we belong to the government of the state of Sao Paulo, but we are also one of the largest manufacturers of immunobiologicals in the region. And we are the main supplier of vaccine and syrup for the public health system in Brazil. We, in example, vaccinate more than a fifth of the population of Brazil. And we are among the 3 largest producers of influenza vaccine every year. So let me go very quickly to this study. This is the PROFISCOPE study, coded code O2IB. This is the pivotal phase 3 study that we established in accordance to the joint clinical development agreement with Sinovac signed on June last year, and we started this study in July. It was a really quick effort from both teams, and this study was focused in healthcare professionals working in services taking direct care of COVID-19 cases. It means that, and this is something that we always advise, that this is something, when you extrapolate the data, we need to pay attention that this is not community population. This is a population that is exposed every day to COVID-19 in a high dose of viruses around. So this is important to notice that this is not a community population, this is a really highly exposed population. And it is also a characteristic of this population. They work with COVID-19 cases, so they know how to recognize a case. And that is one of the characteristics, because they are aware to detect in themselves even the mildest case. The vaccination was stated in a 1-to-1 ratio, as most of the phase 3 clinical trials developed for all manufacturers. And our primary efficacy objective was the virological diagnosis of symptomatic case 2 weeks after the second dose. And this is also an important point to notice. We are talking about disease. We are not talking about infection, as most of the manufacturers in these pivotal studies. Also, we take a look on the primary safety objective following the adverse reactions in the first week of each vaccination. And there are also some secondary objectives that I want to underscore. One is the protection against the severe diseases, so using scoring to detect the severity of disease, and also as a safety measure to assess any evidence of vaccine-enhanced disease that is a concern in general in any new vaccine, and also safety up to 28 days. So this is also a characteristic of this protocol. We actually have only one amendment along the protocol to the obtention of the primary analysis. This is kind of unusual in those phase 3 trials of COVID-19 vaccine that has been disclosed so far. And there is the difference between the one version to the other is mainly because we enlarge the number of participants due to two reasons. At the beginning, we exclude participants with a history of previous SARS-CoV-2 infection, either by a serological test or by history. And also because in the new version, we were able to include elderly people as far as we obtained the results from the phase 1-2 trials in China on the elderly population. And also we changed the design to an endpoint driving protocol. So this is the calculation for this endpoint driving protocol. We have one interim analysis for futility. And so this is the details of that you can see in the protocol. We have published the protocol, so let me see. Here is it. So this is the publication of the protocol. It's in the journal Trial. So who wants to see more details about what is the protocol about, please go ahead and take a look in the trials. This is an appendix to this article. The full protocol is available since October. So the definition of COVID-19 for this trial was very, very wide. We take any symptom with 2 or more days to trigger the PCR for SARS-CoV-2 detection. So even if a person has only a headache for 2 days, it could be enough for having the SARS-CoV-2 PCR. And the reason why is because this population is the healthcare worker population. So we need to be aware that they don't have anybody that can be with a mild disease because they are at risk by themselves and also at risk of infecting somebody else around. So this is really important, and that is the reason because we lower the bar to even the mildest cases. We take all the samples were taken by respiratory swabs and in certified labs or in our central lab here at Butantan. We also take a lot of attention to the events of special interest. There is a list that was created by the Bracton Collaboration Group, and we follow it very carefully, all the cases to be aware that we will not have any problem in terms of these special events of interest. So here in the results, let me show you very quickly the baseline characteristic of this population. As far as we start very late and we are talking about healthcare workers taking care of COVID-19 patients, it was expected to have very few participants in 60 years old or older. Normally we have more than 600, so we can draw some conclusion in terms of safety that were very valuable. We also have most of the participants are female. That is also representing the demographics of our healthcare worker population, and they identify themselves as white, knowing that in Latin America this is not absolutely true. This is just the color of the skin, but it doesn't mean that it's actually the genetic background. There are some people that reported some underlying conditions. I want to underscore that there is a lot of people that reported obesity, and more or less a fifth of the population. And this is important because obesity has been associated with a decrease of immune response in some other inactivated vaccines. So it was good to have a fair representation of this population. This is the flow chart of the study. It's quite complex, a kind of busy figure, but I probably you will see it in more detail. We are preparing the publication, so probably in very few weeks you will get the publication and you can see it in more detail. So just to mention here that we have, of course, one. This is an endpoint driving protocol, so we will have more participants in the safety analysis and less participants in the per protocol population and the intention to treat population, because this is a trial that is still ongoing. So probably after this initial report of the primary analysis, we will have further reports of the ongoing population. In terms of safety, you can see that as comparing the light and the dark bars that represent the vaccine and the placebo group, you can see that they are quite similar. And indeed, it was what was reported by our clinicians, that it was practically indistinguishable, the people having vaccine and placebo. And this is also something to remark, because there are other phase 3 trials that they have a huge difference between the placebo and the experimental arm, so it could allow to biases. In this case, we don't have this problem because those bars are quite similar. And when we unblind the study, what it was really the most important adverse reaction during all the study was the pain in the injection site. So this is also expected, and it is related to the inflammatory processes following the vaccination that will trigger the expected immune response. So this is actually a good sign. There is nothing relevant, and this was nothing that jeopardized the usual activities of the participants. Regarding the systemic adverse events, there is nothing that is remarkable. You can see that the dark and the light blue bars are quite similar in all the parameters. I would like to underscore here allergic reactions were very limited, less than 1%, and those allergic reactions controlled very easily in the ambulatory services with oral medication. We didn't need even intravenous medication for anybody in the study. So this is a signal of the safety of this vaccine. So let me go to the efficacy, and first of all, I want to explain a little bit for those who are not familiar with this WHO progression scale. So this WHO progression scale was stated for trying to standardize how to report the clinical trials, either for prophylaxis or treatment. And it goes from 0, that is a person that has some symptoms, but it has no viral RNA detected, to mild disease. The score 1 is the asymptomatic case. The score 2 represents those who are symptomatic but has no requirement of any medical assistance. And score 3 is still mild disease, but it has some need of assistance. The score 4 and 5 are people that are hospitalized either without oxygen or with oxygen by mask or nasal front. And the score 6 or higher are the severe cases, so with different levels. And what we know is that in the public health perspective, what is more relevant indeed is this, first of all, to avoid the severe disease, so score 6 and higher. And the second point is to avoid the hospitalized cases, so score 4 and higher. Nonetheless, we know that if we want to set up a study to detect only those cases, probably the size of the sample size would be enormous, and the time required to obtain answers would be too long, and that is not feasible in terms of an emergency in public health. So, what we took as a decision was to lower the bar as low as we can. So we even take the mildest among the mildest cases. So it's the score 2, the really, really mild cases. We know that the consequence of that is that we will have low efficacy. It's expected that probably the efficacy was lower for the very mild cases. If we took to the if we want a high score, probably we will have a high efficacy. But the advantage of this is that we will have more cases more quickly, so we can answer this emergency in public health. So this is the decision. So what we want to see in general as the effect of this kind of vaccine is that the vaccine is able to decrease the score in general, to decrease the intensity of the clinical disease. So let's see what happens. So the numbers in red are the ones that had significant statistics in the following slides. And we can see that in the overall, so taking from the score 2 and higher, we have an efficacy of 50.7. You can see that it's a little bit different because we cleaned that we were reporting in early this year, because we have a cleaning of the database, so we can and we have the outcome of some of the cases. So that is the reason, because we have a slightly different number that we have presented before. So this is a significant number. We set up as a bar for a significance lower bound of the confidence interval, 95 in 30%. So you can see that it complies with this, also with this condition. And in terms of what is really, really important, so in terms of score 3 or more, so it means people that has still mild disease, but would require some medical assistance, we will have every 3.7% of efficacy. And for the score 4, meaning people requiring hospitalization, moderate to severe cases, we have 100%. So, and this is also statistically significant. So this is really important, and what we need to interpret this information is that we clearly have a trend. So this is a trend that the vaccine is really accomplishing this effect of decreasing the intensity of clinical disease. That is the effect that we are looking for. So it is really a success that we see that it is a stage of having more efficacy according to the highest score is actually has a logical explanation, is scientifically sound, and it makes us very, very happy because we really prove the efficacy of the vaccine. Regarding the previous exposure to SARS-CoV-2, we really have limited numbers in people with previous exposure, but in the people with no previous exposure, we have a result that is significant. And also, we have the same result for people 18 to 59 years. We have no enough cases in the people with 60 years of age or older to make any statement. Unless we know that for the phase 1-2 studies in China, that the immune response is analogous either in the if you are younger or older than 60 years. So in terms of the interval between two doses, I would like to underscore here that there is a trend of having better efficacy if you have a longer interval. It was not a surprise. We put a very short interval of 14 doses as a target because this trial was designed as an emergency use trial. So we think that for these people that was highly exposed, 2 weeks of difference is a difference. But we currently recommend people to extend to 28 days, and we know that for the phase 1-2 studies in China, that we have a higher titer of antibodies, and so it's probably better to wait a little bit and up to the 28 days between both doses. And regarding underlying disease, there is one that I want to underscore. As I mentioned, we have some concerns about how would be the vaccine, how is the behavior of the vaccine regarding obesity. And we see that it works pretty well. So we are very, very happy to see that in this case, we have no concerns for these inactivated vaccines, that it will be jeopardized because of the obesity. On the other underlying medical conditions, there is no enough number to draw conclusions. And this is the Kaplan-Meier curve of the modified intention to treat results of the study. So you can see that the curves tends to invert. The blue line is the placebo. So, and you can see the very high incidence in the placebo arm in this limited amount of time. And the red one is the vaccine. And you can see that close to the second week, there is a trend to change. So it might be some effect of the first dose. Unfortunately, as far as most of the participants took the vaccine, about that time we cannot draw more conclusions about what would be the effect of the first dose after the second week, but it tends to be that probably there is something that is happening at this time. So, as a conclusion, we can say that the vaccine is safe with local pain as the most relevant adverse reaction, and we achieved the expected effect of decreasing intensity of the clinical disease. And what is most remarkable is that this efficacy was proven in a population with a very high incidence rate. So what we expect is that the people with a lower incidence, with a lower exposure in the community, the efficacy would be far higher. And we can also assume that there is some degree of efficacy after 2 weeks after the first dose, and take this information in account because we will took this information later. But we emphasize that the full 2-dose regimen is required to get the expected protection. And we also noted that probably a longer interval, as what we have authorized in Brazil, these 28 days, might provide some additional benefits. And of course, we will need more effectiveness study and real-world evidence to learn more about this vaccine. But this information was good enough to allow us to the use of the vaccine and emergency use worldwide. So I want to thank all the team that was part in beginning with the participants. We have more than 12,000 participants, more than 700 site staff, people from different teams, CROs, our partners, Sinovac and other partners, DCB, the Research Ethics Committee, our regulatory agency, and Visa, our central coordination team, and the funding was from Fundação Butantan and FAPESP, so our local Brazilian funders. So this trial allowed us to do something that was really, really amazing. So if somebody told me that two years earlier that it could be possible, I would say, no, that's not possible, but we made it. So the difference between these two pictures is 180 days. So this is the pictures between the first participant in the phase 3 studies and the first person vaccinated in the by the emergency use authorization in Brazil. So this is absolutely remarkable, and we know that the results of these trials not only allow the people in Brazil to get access to the vaccine, but also in several, the sense of other countries, and we are very proud to contribute to this vaccine in this way. So we can say that, and I have to say that it put it in front of my friends of Sinovac. This vaccine is not only from China, this is vaccine also from Brazil, this is a vaccine from the world. So let me go for the next question that we want to know about the effectiveness of this vaccine. So we set up another project that we call Project Assay. And this is a project that we want to assess what is the impact of the COVID-19 vaccine on a population. So we want to set up a stepwise clinical trial in order to see what would be the protection of the vaccine in terms of public health. So we select this small municipality in the state of Sao Paulo. It's about 45,000 inhabitants per population, and it's called Serrana. And Serrana, we started work since September in enhancing the surveillance of COVID-19 cases in this municipality. And we established a census of the municipality to determine the cluster. So this is using the social dynamic of the municipality to determine the clusters. So we present this study to the community. The community really was enthusiastic to be part of this study. And we set up the vaccination according to the cluster groups. And we are following the incidence of adverse reaction and incidence incident cases. So this is the puzzle that we built up out of the map of the clusters. And we have 25 clusters in this municipality that they are related to the social dynamic of this population. And we put these 25 clusters in 4 different groups. They are represented by the colors. So we start the vaccination according to the group. So this is very quickly the timeline of the study. So we start studying the 6th epic week of this year, and we are following this schedule. So each group, each color start at each week. So we currently are we finish the vaccination of the first dose of all the 4 groups, and I have a very good news to share in this way that is that the acceptability of the vaccine is enormous. So we have 97% of acceptability of the vaccine. The people really, really want it. The people notice that nobody has any problems, so spread the voice and invite the familiars. And of course, engagement of community leaders has been a key to get this kind of acceptability. And there is also another good news, and this is that the safety of the vaccine was proven at a population level. So we are talking about more than 26,000 people vaccinated, and we have only 1 severe event that was not related at all with the vaccine. So this is something that we are very confident that the vaccine worked very well in this controlled environment. So we're very anxious to finish the vaccination, and probably in a couple of months we can share with you some of the results of the study. Of notice, I want to mention that the P1 variant was detected in this municipality by the same time that we started the study. So in all the region, the P1 variant is occurring at a very high level. So one unexpected result of the study was also would be to answer whether this vaccine can control this P1 variant, because we will have effectiveness evidence with an ongoing P1 epidemic. So this is just to thank you, our colleagues. It's a huge effort, also 100 people working on this, and the collaboration between several institutions to move forward in this regard. So when we talk about these variants, we have 2 variants. That one is a variant of interest, the P2, and other one is a variant of concern, the P1, that has been a real trouble for us here in Brazil. And we have some news. Unfortunately, we have very, very preliminary results, so that's the reason because we cannot disclose more information. But in the wild-type neutralization assays, we saw that there is a very good result. These samples were collected before the emergence of the P1 and P2 variants, so to have more reliability that there is no interference with natural infection. And what we have proven in these very preliminary results is that whenever the serum is neutralizing the parental strain, is also neutralizing the P1 and the P2 variant. And the magnitude of the immune response tends to be more or less the same. This is also something that more or less expected, because this kind of vaccine, when you have the whole virus inactivated, you create a polyclonal immune response that can be more resistant to any mutation. It should be a huge amount of mutation to decrease the magnitude of the immune response, or to interfere to jeopardize the neutralizing response. So this is some are very good news, but this is in the lab. What we need really is the real world evidence. So, but we have some reports of decreasing incidence in the age group that were already vaccinated, and other groups in the same area with P1 circulation, apparently they didn't have this decreased incidence. So let's look at one example. This is the city of Sao Paulo. And so just to show you what is going on, in Brazil, most of the vaccine that is distributed in the country is more than 4/5 of the vaccine distributed in the country is CoronaVac, and the remaining ones are from Oxford. And we have very limited amount of people with the first dose and the second dose. That is the reason, because we will not have huge effects, so we will have a really a huge increase of the disease at this moment in Brazil, as probably you have learned from the news. And the effect of the vaccine is very limited, because Brazil is a huge country, so having enough vaccine for everybody is difficult, and the effect of this limited percentage of population that is vaccinated is also not good enough to be tested in the real world. So this is in general what we are aiming for March for the vaccination groups. And I will show you the example of the city of Sao Paulo. So you can see this is the evolution of the severe acute respiratory infections in this year. And you can see that a huge increase is the effect of, in the first weeks, you can see the effect of the New Year and Christmas Eve. But in the second part, you can see the effect of the P1. So what we are seeing is that actually is an ongoing increase, a very rapid increase with the P1. And in the lower part, you can see where the vaccination occurred, for which age groups. So we started in 8th of February with 19 years old or older, then 20th of February, 85 years old or older, 27th of February, 80 years old or older. And you can see that the P1 is also increasing the number of infections, even in the younger population, 20 to 59, but most of the population that is affected is 60 years or older in terms of these severe cases. That is our focus right now. So we want to take a closer look to the people in the 60 years old or older group. So you can see here what is the behavior of this group. So in general, in most of the groups, you can see that there is an important increase. Probably the only one that has a different behavior is the 60 to 64 years old, and sometimes probably is under subnotification that probably it get this stop, or probably the people is taking more care, or we really don't know. But what is really remarkable are the lines that you can see in the lower part, that are the blue one, that is 90 years old or older, and 85 years to 59 years in the orange one. That you can see that there is a trend that was trying to rise, at some time it kind of stopped. It's a containment. So what we think is that this is something that can be related with the vaccine. So is there an actual difference between the trends in other age groups as compared with this one that are the most vulnerable among the elderly population? So it's kind of like the vaccine, even if they couldn't complete the full vaccination scheme, like 2 weeks later, you start to see that there is a trend of contention of this pressure in the attack rate. So again, is it this 2 week again coming back that I mentioned before? So probably there is some effects from the first dose. And this is some real world evidence that probably this is for real. So you can see this is the combined population. So you can see in the red dot line the behavior of the people with 20 to 59, and you can see that this behavior, of course, the scale are different, but you can see that in this trend is increasing with no stopping in comparison with those groups that start the vaccination earlier. that apparently stopped. So this is a very interesting evidence. We need to follow up, and we need to follow up what happened in particular with the groups that are coming to the vaccination. So we are very interested to see what happened with the people with 80 years old or older, and so on, as far as these cohorts of people is getting vaccinated with the local immunization program. So this is a glimpse of hope, of course, when you see this kind of information. And this is the last slide I want to share with you today. So thank you very much for your attention. I expect that we're passing too much the time, but thank you for your patience.
Thank you, Ricardo. Nice work, and we highly appreciate our Brazil College, a great effort in the clinical studies of CoronaVac in Brazil. And we know, all of us know, currently COVID-19 is still very serious in Brazil, and but our CoronaVac already large used in this country. And also great to see the vaccination start to change the epidemic situation in 90 years old in São Paulo. And actually, we are looking forward to more data from Brazil. Thank you again. Next, I will invite Dr. Sarat from Turkey to sharing the data from another phase 3 clinical study of CoronaVac in Turkey. Professor Sarat, you know, is principal investigator of Turkey trial. He is the president of Turkey's Society of Internal Medicine and Secretary General of Turkey's Society of Hospital Infection and Control, and of International Society of Antimicrobial Chemotherapy. So now I hand over to Professor Yunel. So Professor, you have 20 minutes, and please go ahead.
Okay. Thank you. Dear colleagues, I'm very glad to be here and sharing the results of our phase 3 study. But we just finished the study, so I do not have very detailed report today. I'll just share the mainly the efficacy results, and we are preparing all the detailed results of the study. And it's very similar to what we heard by Dr. Palacio. And I thank him because he explained everything about the study. Actually, we followed almost the same thing. It is a multi-center, randomized, double-blind, placebo-controlled phase 3 clinical study. And it is 2-dose vaccination, 14 days apart. And we use the 3 microgram, 0.5 ml equivalent to 600 standard units per dose. And at the middle of last June 2020, the representative of Sinovac here in Turkey had an agreement with the health institutes of Turkey to run a phase 3 study with the CoronaVac. And that body is the part of the Ministry of Health, actually. And I work in a big university hospital here in Turkey, in Ankara, and I'm the chair of the Vaccine Institute of Hacettepe University. So I've been offered to organize and run the study, and we have started to work at the end of June 2020, and we have written down our own protocol, of course, by getting help by the company Sinovac. And according to the attack rate of coronavirus infection at those days, we have calculated that we need 13,000 volunteers, and the endpoint was the number of the symptomatic cases 14 days after the second dose, and the number for that is calculated to be 40. And we had 2 groups, actually, 2 cohorts. One is the healthcare workers as a high-risk group. We plan to start with them, and we would follow them until 1,000 enrollment. And we have decided like that because we could follow those healthcare personnel in our hospitals carefully and closely in the perspective of the side effects, because it was the one of the first phase 3. So we had concerns about the side effects, decided to go to the healthcare workers first. It's called K1, and it would be a 1-to-1 ratio. And when an independent follow-up group, especially the side effect follow-up group, recognize or help us and permits us that yes, it's safe, then we would progress to the cohort 2 with the 2 vaccination and 1 placebo arm. And the case definition for the 2 days after the second dose, we were expecting to see at least 1 symptoms of corona infection that has been published before, at least for 2 days. And in those symptom positive cases, then we would do the RT-PCR and prove that those symptoms are because of the corona itself. And for the inclusion and exclusion, the main limit, all the volunteers has been tested for PCR and for antibody first day, and we get the results in the evening. If they are negative, then we enroll the patient or the volunteer to the study in the next day. And we have make the applications to local and the central ethical committee. It took some time, particularly the local ethical committee. They have asked thousands of questions, and we could start to enrollment in the middle of September. We got the first volunteer at the 14th of September, and we have organized in 24 different centers all over Turkey. And by February 2021, more than 25,000 doses were applied, including the revaccination of the placebo arms. And we had one extraordinary or unexpected thing during the study. In the evening of 23rd December 2020, I have been asked by the Central Ethical Committee to make an evaluation and an interim efficacy results, because the Ministry of Health was asking, not totally breaking the codes, but an evaluation, what is the efficacy and what is the side effects ratio, because Ministry of Health was ready for the decision to import the vaccine to the country or not. So this is unexpected and extraordinary. We know that, but we had to do this evaluation, and we have studied, those independent group composed of 5 colleagues who are very experienced in the area. They have just looked at the data, how many cases, how many in CoronaVac, how many in placebo arm for the volunteers, and how many cases that I have described to you before, 14 days after the second dose. And in that very preliminary study, there was more than 9,150 subjects screened and 7,370 included, and almost 65 of them in the vaccine arm and 35 in the placebo arm. By the way, in between, up to 1,000 patients, when we have arrived to 1,000 volunteers, all healthcare personnel, the independent group studied the side effects so far, and they have led us to go into the lay people, less risky people. So at that time, at the end of December, the number was almost close to 3-to-1. And as the 23rd of December, all those patients or the volunteers were enrolled from and randomized from 24. And at that time, there was 10,000 doses were applied. 3,457 subjects were in the second dose. And here, this is the Turkey's map. This is famous city, Istanbul, and this is capital, Ankara. And as you see, the 24 centers have been chosen from different parts of the country. and that very preliminary result. We didn't look at any other things, just the COVID positive, PCR positive COVID cases that has been diagnosed 14 days after the second dose. Just look at the numbers, only the numbers. And at that time, there were 1,322 cases who were after 2 weeks after the second dose, 752 of them in the vaccination arm and 570 in the placebo arm. And you see the percentages over here. There was 3 cases in CoronaVac. They all are very mild, and there were 26 cases in the placebo arm. As I recall, at least 5 of them were hospitalized. So out of that 29 cases, the vaccine efficacy has been calculated according to the number in the 95% confidential interval, and with the lower limit of the efficacy, 72% and up to 97% upper limit. The vaccine efficacy calculated to be 91%. I know it's very preliminary. I know it's very unexpected, but to help our Minister of Health, we had to do this calculation. And after that, and here you see the chart for that. This is the 20 days. So 14 days is the second dose. So about 1 week after the second dose, the differentiation starts, as you see over here. And the safety analysis were performed with the data of 2,964 subjects, and there were more than 1,000 events observed in 593 subjects. And the very common ones was systemic ones, fatigue and headache, as you see over here. And after the second dose, even the fewer numbers with the same symptoms. And the most frequent local adverse events was the pain at the injection site, being 0.5% in the first dose and the 1.9 in the second dose. So after that, we had a long discussion because Minister of Health asked us to go on to the study, but some of the group members was worrying about the Helsinki Declaration because we have declared the result. We may not process with the placebo study. And we write back to the Minister of Health, to the Central Ethical Committee, where we would like to stop. And when they have answered us back, and we stop the study, and we make the real code breaking. As you see the dates over here, unblinding for healthcare personnel was started on the 14th of January. And 14th of January is the day the government gave the emergency use of vaccination in the country and started to vaccinate the healthcare personnel. And unblinding was completed at the 22nd of February for the rest of the group. And the updated efficacy results that I'm going to present you today was studied at the 3rd of March as the preliminary result. And again, the same centers. And oh my God, I have forgotten okay. I'll try to find that slide later. Let me finish that here. Again, we use the centers. We have same centers, of course, has gone on to study. We have screened totally more than close to 12,000 people, and the 10,216 subjects received at least 1 dose of product, and the 10,030 subjects received the second dose of the product. And here you see the centers from different part of the country. Some of them has gone up to close to 1,000 cases, and some of them could give only the 100 cases. And in the perspective of the gender and the age, and I've forgotten to say this study included only the people between 18 up to 59 years of age. We don't have over 60. And as you see, the numbers among the genders and the age is quite equal. And I do not have the other detailed data of the different groups available now. And very basically, vaccine efficacy, but this time calculation is different, of course, because in CoronaVac arm, we have 6,559 cases, and the exposure of them are different. So the person year exposure is 283.89, and placebo arm, we have 3,471 cases, and the year exposure is 166. Here you see the incidence in both arms, and we stopped at the 40 cases actually. 2 weeks after the second dose, we have 41 cases, 32 in placebo arm, and 9 in the vaccine arm. And this is the first endpoint, vaccine efficacy against symptomatic infection. Again, the person will have at least one symptom, at least for 2 days, and will be PCR positive. And the total cases that we have evaluated for this one was 10,030 cases. And in this calculation, the vaccine efficacy was 83% 5, and the efficacy lower limit is 65 and up to 92 in the 95% confidence interval. And the other endpoint was the avoiding of the severe infection. We described that as the need for hospitalization. Of course, there are different severity level in the hospitalization, but this is the general evaluation of the hospitalization. In the vaccine arm, there was no cases who has to be hospitalized because of that corona infection. And in placebo arm, there were 6, and because of the 0 to 6, we had 100% efficacy for the hospitalization. And when we do the chart again, here we have the 14 days, as you see. About 1 week after the second dose, we start to see the difference, and the protection is coming out. But with the first dose, everybody is so eager in the country. What is the protection level after the first dose? We are doing those calculations, and we have numbers for that too, but it will be released in the publication. But obviously, the trustable side is the at least 1 week after the second dose. And for the follow-up for all study group, follow-up after the randomization, you know, the rule says you got to follow at least 60 days. And in both arms, we came up to 70 days. And after the 14 days, after second dose, we have the follow-up 42 and 43 days. And since the mass vaccination is started in the country, we had to stop the study. So the follow-up for the 14 days after is 42 days. And for the safety results, adverse events in the first days after the first dose, in the vaccination arm, 6,648 and 3,568 in the placebo arm. No major difference actually. Pain in the inoculated sites and the redness in the inoculation site is not very different than placebo, as you see. And in some systemic effects, even in the placebo arm, we had more numbers, like fatigue, headache, muscle pain, fever, diarrhea, shivering, nasal congestion, and cough. So we didn't have any major safety issue after the first dose. And after the second dose, you see the numbers are almost the same. Local side effects and the systemic side effects you see over here. And the serious side effects, those are the total numbers actually. Among all those 10,000 people, there was only one mild anaphylaxis, I would say, real allergic reaction. She was a pediatrician in Istanbul, and they have hospitalized her for observation for 1 night, but that was not a major issue actually. And the other unrelated issues over here you see. So we did not diagnose any major side effect. So at the end of this 4-month study, I would say, including the first preliminary and then at the beginning of this month, the second preliminary results, I can say that this vaccine, CoronaVac vaccine, is safe and quite effective in the prevention of the symptomatic cases up to 83%, and in the prevention of the avoiding hospitalization because of this infection. And after me, my colleague from Ministry of Health will talk about the mass vaccination. We have gone up to close to 13 million or 14 million doses already being used in the country. And she will talk about the results of this vaccination. Nowadays, the thing that we are discussing, actually, just added, Turkey is using Sinovac and the very limited BioNTech Pfizer vaccination. And comparing to the other countries, we are trying to do our best like all the other countries, and we are in the first 5 or 6 level in the number is the number of the vaccination. And among those 10,000 people, actually, in the protocol, it was included up to 2,000 people would check for the antibody level. We have done it. We are studying we are working on the results. And among them, minimum 1,000 of them would be evaluated for the neutralizing antibody. It's almost finished. And again, among them, about 300 people, we plan to check for the existence of cellular immunity. Unfortunately, I do not have the clear data yet, finished data yet, but in the discussion, I can share some very preliminary results. And the other issue in the country we are discussing, and the effect of the variants, mutations, as Dr. Palacio says, in Turkey, during these days, possibly we are starting to the third attack, because in the last 1 month, the new cases number, daily new cases number has increased from 3,000 up to 20,000. And for the main reason for increase is the problem obeying of the rules, mask distance, and the hand hygiene, and the general rules that has been published by the Minister of Health, or the new, what we call British variant. It came up almost to 50 to 60% of all isolates. So it's definitely transmitting more, so increasing the number. And we had the chance to check the efficacy of 0-14 days vaccination, as we have done in our study, and in the country we have used 0-28 actually. I can explain in the question session why we have done so. And the sera of the people who had the disease, and comparing all those, we can say that this vaccine looks like protecting against the British variant as well. But we did not have chance to check it for the South Africa and the Brazilian variants yet. But thanks to God, they may be problematic. As you see over here, some of the vaccines were not effective against them. The number is not very high in the country yet, and I hope we'll be able to keep it like that. So it is exactly 20 minutes. I'll stop here. Thank you very much for your attention, and I'll be happy, very happy to answer any question you may have. Thank you.
Thank you, Professor. Actually, it's a very encouraging result indeed. Well, unfortunately, because the limitation of time, we did not set up the Q&A section after each speech. So based on this good result from Turkey, actually, CoronaVac was granted emergency use approval in January. And the large vaccination campaign already triggered after this approval. Now, more than 10 million doses of CoronaVac already used in Turkey's population. And today, we also invite Dr. Berak from Ministry of Health of Turkey to sharing some vaccination campaign data with us. Dr. Berak Boro Basura is a medical doctor, has experience in public and private health sectors as a field physician before she had started her career in Ministry of Health of Turkey. She has a long career in Ministry of Health since 2002 at many levels. She has been holding the head of Department of Statistics position in the General Directorate of Health Research from 2010 to 2018, and General Directorate of Health Information System from 2019 until now. Let's welcome Dr. Berak. You have 10 minutes, please. Thank you.
Thank you very much. Okay. Just a minute. Sorry.
Yeah, it's coming out.
Sunuma getirdim mi olacak herhalde. Ha? Tamam böyle. Böyle devam et istersen, gözüküyor.
Yeah, I think it's okay. You can use this model to present.
Okay. First of all, I want to thank you for inviting us to this meeting, the webinar. Today, I want to give you some information about the Turkey's COVID-19 vaccination program. This is the presentation plan. I will give you some information about the priority groups of Turkey and how we make an appointment and what is the vaccine mobile system. And also I will give you some statistics. Today, the vaccination program and the development of vaccines are very important, and also it is the key focus for the ministries to respond to the pandemic. Our ministry and also the science board determined the order of groups to receive the COVID-19 vaccine by evaluating the risk of exposure and the risk of having the disease with the severe courses, risk of transmitting the disease, and the negative impact of the disease on the functioning of social life. This is the list of vaccination group in Turkey. In the first stage, we have healthcare workers and also students in the faculty, medical faculty, and also the social care workers and residents at the elderly care homes, rehabilitation centers. And also in the third part, we have 65 and older people. The vaccination has been completed in stage 1 group. And also in the stage 2, we have frontline essential workers. Still vaccination is going on in this stage. And also we have stage 3 and 4, individuals with chronic disease and other groups below 49. I would like to briefly talk about the appointment system in Turkey. The person can check the priority group status from the e-Pulse, let's say in English, account by the text message. E-Pulse is a personal health record system. You can see all the health information and access your medical history. In case of being among the priority group, appointments can be made from the family doctors or all hospitals. In the health institution, the person is registered with the Aşıla application. Aşıla means vaccinate in Turkish. It is the mobile information system used while administering the COVID-19 vaccine. In the Aşıla, we have QR code camera. It is the camera of the phone. QR code of the vaccine is read by the mobile phone, so we can check the cold chain of the vaccine. We have a very QR code-based cold chain quality system. It's a vaccine tracking system, and we can check the temperature and the stock level of the vaccine in each level. After the vaccine is ready for the vaccination, the location, place, and the method of application of the vaccines are recorded in the system. And if the QR code is say that it's safe, the vaccination is applied. If it's not, system warns you. After the vaccination, all details of the vaccine sent via text message and can be seen also in the ePulse account. So you can have the vaccine card both in Turkish and in English, and can be downloaded and can be used while going to the other countries. I will give you some statistics about the national COVID-19 vaccination program, and also I will give you some international comparisons. This is the data till 18th of March, but today it is almost 13.5 million. Unfortunately, it's 12.3 million because we have to give the presentation till Friday. That's why it's a little bit lower than the real number. And the number of doses are divided by the risk groups. The people who have received at least one dose, it is almost 8 million, and fully vaccinated people is almost 4.4 million. Here in this graph, you can see the people who received at least one dose by age groups. As you can see, it is above the 60 and over ages. We have more vaccinated people than below 60. For the healthcare workers, it is a little bit different from the total population because they are younger, and then the age is between 20 to 60. And this is the social care workers, residents at the elderly care homes, and people with the special needs. The age is distributed in all groups. This is the number of 65 and over people and their partners who receive at least one dose, because in this group people were visited at their homes, and those living at the home and taking care of these people were also vaccinated, even though they were not included in that group. That's why we have under 60, we have a little bit vaccinated people. This is the total number of vaccination doses administered by time. As Dr. Serhat Ünal said that the interval between the first dose and the second dose is 28 days. So you can see after the 28 days, the second dose are also started. The distribution of adverse effects is very similar with the clinical trials. These data are coming from hospitals and doctors reported as vaccine related. The fatigue that affects daily life is the first in line, and the headache also in the very similar number frequency. Acute allergic reactions, gastrointestinal system symptoms, and the fevers are also the adverse effects. I will put the diarrhea, although it is in the gastrointestinal system symptoms, because it seems higher. That's why we put them. We put it in the graph separately. But when we add to the 10%, it will become the first line. And among those who have vaccinated, around 8 million, which have who has at least one dose. A total of 4,300 people experience an adverse effect. It is 52 per 100,000. And I must say the 45% of people with these adverse effects having comorbid diseases, and 19% have allergy, and 40% were using medicine. And 1.52% per 100,000 are needed to be hospitalized, and also 1.19 per 100,000 experience a life-threatening situation. When we compare our results with the other countries, it is also the data are coming from our World in Data. The distribution of the dose is very different in countries by countries. For example, in United Kingdom, first dose is 93% of the all doses. In Turkey, it is 65%. For example, in Canada, it's 82%. And when we look at the distribution, the percentage of the population, 37% of the population is vaccinated in United Kingdom, and it is 9.5% in Turkey. And this is the comparison of people receiving at least one dose per population and the total number of doses. Here you can see the population percentage and also the number of doses. This is the comparison of the full vaccinated people per population. In Turkey, it's 5.2%. It's higher than the other countries. And also, although the number of vaccination is almost similar, the percentage of population is higher. In Canada, it is 1.6%, and in United Kingdom, it's 2.6%. In this slide, the proportion of people, you can see the proportion of people who received at least one doses. Although the distribution, the cumulative trend is high in United Kingdom, and other countries are almost the same. When we look at the fully vaccinated part, it's almost not equal with other countries. You can see the United Kingdom is lower, and for example, in Turkey, it is going higher and higher in every day. But in Canada and Italy, it is going also higher. Thank you. Shishan.
Thanks, Dr. Barak. And actually, as the vaccine manufacturers, it's really fantastic for us to see this large vaccination campaign. And we are also really looking forward to working together with the colleague in Turkey and to win the battle finally against this COVID in Turkey. So next, since this is online webinar, so we don't have a COVID break, but actually there is, I think there's 20 or 30 people here. So I suggest, because already the COVID break arranged in the corridor, so people sitting here can go out and to have coffee, but we will go on for the sections online. All right, so next, I would like to invite Dr. Kusnandi Rusmil from Indonesia to sharing the data of the trial in Indonesia. Dr. Rusmil is the principal investigator of the phase 2 clinical trial for CoronaVac in Indonesia. And he is also the member of the research unit of the School of Medicine in the University of Padjadjaran. And so he is leading a team and to a lot of research, not only for COVID-19 vaccine, also for hepatitis B, OPV, influenza, and etc. So Dr. Rusmi, please start your presentation. You have 20 minutes. Thank you.
Yeah. Okay, thank you very much. I want to share screen, please. Okay.
Yeah, looks nice.
Thank you for the opportunity join the corona vaccine clinical trial. I am very happy. And this moment, I want to present about the inactivated vaccine COVID-19 clinical trial in 18 years until 50 years of age. The clinical trial conducted by Faculty of Medicine University Pajajaran, collaboration with Bio Farma and Sinovac Life Science. Yeah. This is phase 3, observed blind randomized clinical trial for the inactivated vaccine, and this is a preliminary result. This slide describes overall study plan we need for the efficacy 1,020 subjects for efficacy, safety, and immunogenicity. For the efficacy, we divide two group vaccine group, and the vaccine group were divided 3 groups for batch consistency. We saw that the batch consistency, lot 1, lot 2, lot 3, and the next. These slides describe overall study and plan. This is a flow chart. Before the study, the subject must be done for PCR and rapid test for COVID. If the negative, the subject can join the research. This is according for ethical committee, because many of the ethical committee very it's the new disease. She is very careful about that. That's why before the study, the subject must be negative for the PCR and rapid test for COVID. Before vaccination in visit 1, blood sampling was carried out for serology. And then after 4th day, the vaccinated subject have the vaccination after 14 days. Then after 14 days, the second vaccination in the visit 2. Yes, you know. And then blood sampling was carried out for visit the visit 3A, 3 months after visit after visit 2 plus 14 days, and then visit 4 after 6 months after visit 2. Blood sampling was carried out for 3 months and 6 months. COVID monitoring was conducted after the first dose of vaccine by combination of active follow-up monthly by monthly phone call and text message and automatic reporting. COVID-19 monitoring by nasopharyngeal swab for the suspected case after 14 days, second dose. Yes. Nah, inclusion criteria is the clinical healthy adult age 19 until 50 years. Subject has been informed properly regarding the study, signed the informed consent. Subject commit to comply with the instruction of the investigator and the schedule of the trial. And the exclusion criteria, subject concomitantly enroll or scheduled to be enrolled in the adult trial and contact with novel coronavirus infection person positive for nucleic acid detection within 14 days prior to trial. Evolving mild, moderate, severe illness, especially infectious disease, and history of asthma, allergy of vaccine, history of uncontrolled coagulopathy, blood disorder, and patient with serious chronic disease, subject who have any history of confirmed suspect immunosuppression or immunodeficiency, the previous 4-week treatment, and subject who has history uncontrolled epilepsy, subject received vaccine within 1 month before of immunization. This is the primary result. The primary result was based immunogenicity data analysis for the first 540 subjects in immunogenicity subject until 14 days after the second injection. Safety data analysis for the first 540 subjects in immunogenicity subject group until 28 days after the second injection. Efficacy analysis from 1,620 subjects with cut-off data 5 January 2021. This overall about the randomized 540 subjects. In the randomized, we have 3 batches of vaccine, batch A, batch B, and batch C. Batch A 135, batch B 135, batch C 135, and before injection, early termination for batch A, and batch B 1 and batch 3, and none. Is that? Yes. And this is the demographic data of 540. The vaccine mean age mean age 36 years in vaccine, and placebo 35.3 years. Mean height 161, in placebo 161. Mean weight 64 kilogram, and placebo 65 kilogram. Male 56, and placebo 63%. Female 43%, and placebo 50%. This is the IgG antibody titer with ELISA in V1 in visit 1, visit 3, and visit 3A. In visit 1, seropositivity rate we have 44 subjects with positive in vaccination before injection, and the placebo is 14, but this is not different. GMT 202 20 and placebo 220. After we give injection 2 times, the seropositive increase 99.74% and placebo only 5%. Yeah, the range 19.2 until 100%. The seroconversion for IgG and GMT 5,181 and placebo is very different. Yeah, and after visit 3A, after 14 days, the concentration is decreased from 19.7 to 19.23, and placebo is different. Yeah. The incident of laboratory confirmed COVID-19 case within 14 days after the second dose. The primary outcome of the study was to assess the efficacy to dose of SARS-CoV-2 vaccine in preventing COVID-19 cases compared in placebo. This was measuring using symptomatic case laboratory confirmed of COVID by real-time PCR testing from 14 days until 6 months following the second dose. By the date in this interim report, we were 25 symptomatic laboratory confirmed COVID-19 from acting following second dose in 3 months. This vaccine symptomatic confirmed in vaccine group 7, in placebo 18, incident rate per 1,000 0.039, and incident rate per person in placebo 1.1120, and then the vaccine efficacy that by 65.3. Yeah, this is the immunization seropositive rate in the graphic. In visit 1, yeah, this is a vaccine and this is a placebo. IgG, the vaccine is high, more 19.7, yeah, and placebo is very low. In neutralization antibody, yeah, and before injection, no, and after injection is high, yeah. The seropositive rate of IgG antibody using ELISA assay after 14 days after second injection was 99.74 compared of 5.29% in the placebo group. Oh gini nih, mudah-mudahan. I'm sorry. Yeah, the second injection was 95.70 compared 0.75 in the placebo. This is the immunization seroconversion rate. IgG antibody seroconversionnya most of 100%, yeah, 7 97.48 compare 0.70. Seroconversion for neutralizing antibody after 14 days. After second injection, yeah. This is the ELISA GMT. You see, GMT, yes, the neutralization antibody. Adverse event according onset of time: local pain, redness, induration, swelling, fever, fatigue, myalgia, unsolicited. You see that the local pain within 30 minutes, 0 until 7 days, and 8 until 28 days. We see that the vaccine, and this is placebo, vaccine and placebo, vaccine and placebo, yeah, and vaccine and placebo. We see that, yeah, not different. In conclusion, the inactivated SARS-CoV-2 vaccine is tolerable, immunogenicity, and has 55.30 efficacy against COVID-19 in healthy adult age 19 until 59 years with 2 doses scheduled of 14-day interval. This is emergency use authorization in Indonesia in January 13, 2020. That's why we use the vaccine Sinovac for all the senior people and for the health worker. Thank you. Okay, thank you very much for your attention.
Thank you very much, Professor. And we highly appreciate this great work made by our Indonesian colleague. So next, I would like to invite Dr. Alexis Kalergis to sharing the current process of the clinical studies conducted in Chile. Dr. Alexis is full professor and director, Melanie Institute on Immunology and Immunotherapy of the Catholic University of Chile. Actually, we know each other since 2019, and previously we are in a discussion on some novel vaccine collaboration. So when COVID-19 happened, we quickly decided to conduct a phase 3 trial for our CoronaVac in Chile. And currently the trial is still going on, but already have some data available. So let's welcome Dr. Alexis. Doctor, you have 20 minutes. Thanks.
Can you switch allow me to present from the picture, not from the camera?
Yeah, we can see you quite clear, but the voice maybe have problem. I think you have 2 devices switch on, right? Maybe you need to turn off one of them. The slide is very nice. But you are mute. No voice. No voice.
Can you allow me to use my microphone from the picture?
Yeah, no, I can hear you. Yeah, also I can see your slide, the first picture.
My microphone on the account is with the picture, not with the video.
Oh, but I didn't see the picture of microphone. I think you can go ahead with this. But no voice.
My account with the picture, you just need to activate the microphone, please.
You mean? Say again. Because the voice is too low. Hi, Alex. My colleague told me you have a 2-account login, so maybe you need to turn off one of them.
On the account with the picture, please.
What picture? You can text me a message.
So I'm in 2 accounts. If you could just turn on the microphone in the account with the photo.
Okay. All right.
One second, waiting. I have to make a little change in the computer, okay?
So, Professor, my colleague informed me you log in 2 accounts. So they need you turn off one of them.
Okay, can you hear me now?
I think you can go on now. We can hear you and also can see your slide.
Let's see if it works. Okay, thank you, Wayne and all the colleagues from collaborators from Sinovac. I will just describe in my 20 minutes the little history behind the collaboration, as well as the data from safety and the immunogenicity from the trial in Chile. COVID-19 is a big issue everywhere, so collaborations are needed. We early realized that drugs and vaccines will be highly demanded by everybody. So international collaborations are required in order to accomplish successful vaccine use. And the different approaches have been developed to create vaccines against COVID-19. We've been working in Chile for about 20 years on vaccines development for respiratory viruses. So we met with Sinovac earlier before the pandemic in order to collaborate in the development of other vaccines. But when COVID-19 came up, then we started working together on COVID-19 vaccines. And the focus of the antigens for this virus have been several. Most people have focused on spike, but it's important also to think on other antigens such as E, M, and the nucleocapsid protein. And we are in Chile working as well on research on a vaccine that considers these antigens. And one of the reasons for us to collaborate with Sinovac was the fact that the vaccine was a whole virus vaccine that contains all the antigens. And I'm an immunologist, so most of the problem due to COVID-19 or SARS-CoV-2 infection is an enhanced inflammatory disease. So a vaccine needs to drive the immunity towards the right polarization mode in order to get enough cytokines, T cells, and antibodies, but not an enhanced activation of the immune system. So some of the folks in the lab working on both the trial as well as the vaccine development that we're doing in Chile. Now, this has been important. I mean, everywhere COVID-19 is a problem, but it's important that we can also enhance our capacities in Latin America in terms of vaccine development, both from the basic science and the clinical research. So through several years, we have established collaborations with many countries, USA, France, and recently with Israel and China. And also internally, we have organized as a consortium of universities and hospitals that want to test different vaccines during the pandemic through clinical trials. So it's been a collaborative work in Chile and towards other countries. And the collaboration with China was initiated thanks to an international immunology meeting that took place in 2019, where we presented our results on the RSV, respiratory syncytial virus vaccine. So we have published in several papers. And we met Sinovac thanks to people connected to the Chilean embassy in Chile. And this was, October, a couple of months before the pandemic initiated. But led to the opportunity to know Sinovac, and especially we knew them in Chile, before, because some of the vaccines, such as hepatitis and influenza, had been previously registered by our regulatory agency. So the Sinovac vaccine was one of the leading candidates, and the data that was published in Science was very, motivating, and it's an inactivated virus vaccine, and the preclinical data, as well as the phase 1 and 2 that they registered in clinical trials, led to 2 publications, 2 in Lancet, for adults between 18 and 59 years old, and the elderly, and the 2 publications showed safety and immunogenicity. So we established this agreement that is aimed to scientific collaboration with the possibility of accessing 2 doses for massive use in Chile if the vaccine was eventually approved for emergency use. So a year later, I just realized preparing this talk that Sinovac visited Chile. It was also October 2020, and we met with the authorities of the government in order to promote the carrying out this major trial, and as well with the authorities at the university, because we consider this collaboration as a long-term collaboration. So since we established the collaboration agreement with Sinovac, it took a lot of work just to put this together in Chile, from the legal, the regulatory, the communication, the clinical, and scientific elements of the study. So we have made progress in all of them in terms of putting this trial together. It's a lot of work for carrying out a phase 3 at the university together with hospitals. And we were able to successfully present the study design to the ethical committee and as well to the Chilean regulatory agency, which is a grade 4 agency as the FDA and the EMA and Anvisa in Brazil. So everything went well in terms of the preclinical data, the phase 2, and of course the GMP manufacturing. Can you hear me well?
Yes, very well. You can go ahead. Well, I think you're mute again.
No, can you hear me well? Is a slide there?
Okay, you come back. Go ahead. Yeah, I can hear you.
Yes, we see the slide.
Okay, can you hear me?
Yeah, I can hear you.
All right, so the trial was approved in September 2020, together with another clinical trial in Chile, and we registered the trial in clinicaltrials.gov, which is important. And this is the rationale for the trial. We had 2 stages, and I will explain that in a bit. Total number of enrolled volunteers was 2,305. We have about 30% of the volunteers were over 60 years old, and we had 2 different vaccination schedules. 48% of the vaccine of the volunteers received 0-14, and 52% 0-28 days for the dose 1 and the second dose. And so far, about the 2,305 volunteers, we have registered 20 cases of COVID-19, which is kind of low. I mean, the virus was not circulating very heavily when we started the trial. Now it's actually going up. After second dose, we have only 6 COVID-19 cases, and 5 of them are in the 0-14 vaccination schedule, and only 1 was observed in the 0-28 vaccination schedule. So it's a low incidence, at least in our study, for COVID-19. Out of the 20 cases, we have only 1 hospitalized case of one of the subjects. I will just tell you, this is a very it's a multi-centric study in Chile. Several universities and hospitals, both in Santiago as well as in another region, which is about 100 kilometers away. So a total of 8 centers participated, and we had 2 stages of the study. First, stage 1, which was aimed for health personnel over 18, and most of them received 0-14, and we were at that moment using placebo because there was no vaccine when we started approved for emergency use. And in stage 2, we opened the study to general population, and because the emergency use vaccine was there, according to the discussion with the ethical committee and the Chilean regulatory agency, we decided just to stop using placebo and just focus on the immunogenicity and comparing 0-14 with 0-28. So this is the overall structure of the study. So about 50% are in the 0-14 schedule for vaccination, and in both, and about 50% as well are in the 0-28, and a little bit more in the 0-28. And we have people, volunteers under 60 and over 60 in similar ratios between the two different stages of the study. And I won't go too much into detail about the safety because it's been discussed extensively so far, but we have similar results that was shown in Brazil, Turkey, and Indonesia. The adverse effects for this vaccine are rather low, and some of them, as you can see in this slide, are compared between placebo and the vaccine group when we had placebo, and also between the first and second dose. And the most frequently reported side effect is headache and very, very little fever, for example, as an adverse effect. So in general, we have very low local reactogenicity. Local pain is about 55%. Most of the effects are mild, 97% mild, short duration. And we observed, and this is something that we have discussed quite a bit, is there were fewer events in volunteers over 60 years old and reduced pain in volunteers over 60 years old. Then we focus on the study of the immune response, and I will probably extend more on this part. We have been taking samples, of course, before the first dose, after the second dose, and then we have data up to day 42 after the first dose. So that would be 28 days after the second dose. And we looked at antibodies against spike by using a certified kit for looking at those antibodies. And as shown here, we have some responses after the first dose, which is day 14, and a pretty good response day 28, which is 2 weeks after the second dose, 14 days after the second dose, and the antibodies remain high up to 42 days. This is the last data set we have, which is 28 days after the second dose. And this is the total. On your left hand side, we have the total vaccinated, and on your right hand side, we have the people over 60. So we do see an equivalent amount of antibody titers for total study comparing with the population that is over 60. And here is the table that shows the total sort of seroconversion. So we find that after the first dose, seroconversion is kind of low, but after the second dose, we do see that most people receiving the 2 doses of the vaccine are positive for antibodies, and the antibodies remain high after 28 days post the second dose. Now we are doing 3 different assays for neutralization. I'll show you some of the data. We're still working on this. We have a lot to do, but basically we do see neutralization capacity at 14 days and 28 days, which is day 28 after the first dose and day 42 after the first dose. We see pretty significant amounts of neutralizing antibodies in the vaccinated volunteers. And again, we get equivalent results for the populations that is over 60. And so in terms of seroconversion and neutralization, we have we find a pretty good correlation. And this is using a surrogate viral neutralization test or kit from GenScript. As I said, we've been doing this different ways. and I'll show you data resulting from a collaboration with colleagues in the university. They have a HIV-based serotype virus, and we gave them our samples, and they found pretty much the same observations. So day 28, which is 2 weeks after the second dose, and day 42, we do see significant neutralization capacity of the serum, which is actually very encouraging. In terms of T cells, and before I go to T cells, just to tell you that we also measure antibodies against N, the nucleocapsid of the virus, and those were rather low. We did not very strong anti-N antibody response, but we're still, looking for. We're still completing the analysis, but the preliminary data show that the anti-N antibodies are rather low. They are there, but they're lower than anti-S. Now, for measuring T cells, we used the mega pools that were provided to us by La Jolla Institute in California, and we measured. We did several measurements. I'll show you today just data from ELISpot analysis, and we found that both in total adults between 18 and 59, we can detect T cells that produce gamma interferon, and the numbers are looking promising after 28 and 42 days from initiation of the study. But most of the T cells show up after the second dose. And so this is the data, and then again, we see similar trends between adults between 18 and 59 and those volunteers that are over 60. And this is some of the same data. It's easier to see. Detecting T cells using ELISpot is possible, and these are primed by this mega pool system that was developed by Alessandro Zeri and his team in La Jolla. So, preliminary conclusions we have, as we have seen, the vaccine is well tolerated, has lower reactogenicity. We have reduced severe adverse events and no withdrawals from the study. We have reduced or non-anaphylaxis. And in terms of the immunogenicity, we can detect antibodies in over 90% of those people vaccinated after the second dose. And we can show that the antibodies neutralize the wild type strain. We are planning to do the variants. We have so far looked only at the wild type SARS-CoV-2 strain. And we can also detect T cells that are specific for the mega pool provided by the group in La Jolla. So T cells and antibodies are there 2 weeks after the second dose of immunization with Sinovac. So just to finalize, it's been a big effort just to take this vaccine into human use and population use. The collaboration is being supported by of course, the science between China and Chile, also by the several ministries of health, science, and foreign affairs, as well as the private sector in Chile by CDC. And it's important to say that during this process, the Chilean regulatory agency visited China and made an audit to Sinovac that was took a couple of days. And right after that audit, a month later, the vaccine was approved for emergency use in Chile, which is, is the result of a month of revision of data derived from different studies, from the inspection as well as our preliminary data. And that led to the initiation of a campaign in Chile. The arrival of the Sinovac doses allowed the process to start, and has set Chile as one of the countries that is leading the vaccination process. And so far, we're still waiting to know how the vaccine is behaving in the population. It's a bit early to tell. There have been, I think, about 2.5 million that received the second dose. Here on the right is a little trend in terms of what's been going on. It's been discussed here that there's a slight fall for the hospitalization of elderly, but still too early to tell, so we have to be careful with this analysis. That is the data that are being collected in the country. But it's encouraging, but still it's a lot to study, and we have to be very careful with the conclusions. It's still too early to make conclusions. So just to finalize, to tell you, it's a big effort between citizens, basic research, clinical research, public sector, government is being really strong, and the private sector was critical to initiate the process, and of course the international collaboration in order to reach to this kind of work. And just to I'd like to acknowledge the leading group. So Dr. Abarca, she's the head clinician in the study. Dr. Bueno, she's the head scientist. Dr. Gonzalez Pablo, he's the executive director, and Dr. Jose Vicente Gonzalez, he's our regulatory person leading to the technical components of the connection with the regulatory agency. Several teams, in the lab as well as in the clinics, and of course, all this work is very collaborative and requires a lot of interaction. So thank you so very much, Wayne, for the opportunity to present.
And thanks, Dr. Alex. And there is really great work from Chilean colleague. Actually, we are also looking forward to more data from this trial. Obviously, this is significant to evaluate this vaccine. And so next, I would like to invite Dr. Rafael to present some real-world vaccination data in Chile. As Alex already explained that, actually this CoronaVac already got emergency use approval from Chile in January. So currently already a large vaccination campaign conducted in this country. So we also invite Dr. Rafael to give some data about that. And Dr. Rafael Bralic is a physician scientist with a strong background in clinical infectious disease, epidemiology research, and microbial genomics. And from May to November 2020, he was the chief epidemiologist at Ministry of Health of Chile. So next, let's welcome Dr. Rafael.
Okay, thank you very much. Hello, everyone, and thanks for inviting me to present today. It's an honor to be here, to be able to share our experience and to learn from what other countries are actually doing. It's really nice data what I've seen today. So the idea is to show you a bit of an update of what we are doing in terms of our immunization program, but also I want to put everything in context and talk about a little bit about the situation, the COVID situation in the country. It's really interesting to understand the moment in which we are vaccinating our population. So it's really challenging for the vaccine. and also to discuss briefly how we are approaching real-world efficacy data. I have no conflict of interest so far. This is an epidemic curve, and you can see on your left the national curve. In blue are actual cases, and in red or orange, you can see what we estimate, considering the diagnostic delay. So as you can see, over the last week, we've been seeing around 6,000 cases every day, which is a lot and compares to our worst peak last year. If you see the tail of the figure, you see that maybe we are entering a decelerated phase that might be concordant with all the non-pharmacological interventions that have been put in place lately. So hopefully we're going to get better. And on the right, the idea is to show you the dynamic of the pandemic over time in each of the country's regions. And as you can see, it's been asynchronous, except for the last 1 or 2 months in which all the regions are experiencing significant outbreaks. Except, and this is an interesting observation, the one that you have on your lower right corner, the Magallanes region, which is probably been the most affected one by the outbreak, and also probably the one that has received more vaccines proportionally. And it's the only one that is going down. So I know it's just an ecological observation and it's a correlation, but it's encouraging. So these are effective. As you can see, it's very concordant with the estimation of cases that I showed you in the last slide. And as you can see on the right part of the plots, the curve is going down, and hopefully that's a real signal that transmission is going down according to the measures that have been taken. So this slide is important. I'm showing you as a heat map the incidence of active cases by age groups. And as you can see, extreme age groups, more than 70 years old, and the very young population has lower incidence rates than those between 30 or 25 to 79 years old. And as you know, might know, our vaccination campaign started from the elderly. So again, this is only correlation, but it's encouraging, and we really need to add a control group to see if these effects can be attributed to the vaccine, or is just that the elderly population learned how to take care of themselves. This is again the same figure of the epidemic curve at the national level, and the idea is to show you how are we doing in terms of our sampling effort, our testing efforts. And in the line shows you the positivity, and as you can see in the horizontal black line, that's the 10% threshold. So the current peak is occurring with a very low, at least low positivity rates, which tell us that we are testing a lot. So our assessment of the number of cases is closer to the real number of infections, and that's good for our tracing and isolation efforts, and also to assess the effectiveness of the vaccine in the real life. And this last slide is just to show you that the proportion of the population that is confirmed with a PCR within the first 72 hours after symptom onset. So we're now around a 50% number, which is not as good as we would love, but it's okay, and it hasn't getting worse, even though we're doing around 80,000 RT-PCRs every day, which is a lot for Chile. So about the program, we are vaccinating with Sinovac and Pfizer. So are you hearing me okay? Because I have some echo.
Yeah, no problem.
Okay. but almost 98% of the population have received Sinovac vaccines. So in terms of the number of doses administered, we are close to 8.5 million doses. Number of people vaccinated with at least one dose is 5.5 million. People that received the complete vaccination schedule is around almost 3 million people. And all of these out of the objective, which is 15 million people within the first 6 months of the program that started massively on February the 3rd. So the full coverage of the vaccination program is around 20% so far. And on the columns in the figure on the bottom, you can see on the table, actually, you can see the distribution of the number of people that have received vaccines according to the region they live. And as I said, Magallanes region is the one that is being more vaccinated with almost 50% of the population. And two regions in the very north, these Antofagasta and Arica, are those that are, with the lower rates of vaccination, around 30%. About the rollout, just I want to show you when we really started the program. So these are probably, the early efforts with Pfizer and some of the Sinovac that, came here before a year. But the program really started on the 3rd of February and started very, very intensively, with the weekend effect, of course. And it went down because, the first vaccinated group was according to age. So as long as they were becoming vaccinated, the rate of vaccination went down, but it went up when, second doses started and when we added chronic diseases to as a second prioritization criteria. And in the bottom, you can see the same, but only with Sinovac. So we're vaccinating a lot. So this is the rollout by sex. Very, very well balanced. Probably a bit more women than men have received the vaccine. And this is the achievement by age group, and unsurprisingly, those that are older than 61 have the most significant advances. And as you can see, one dose has been received by more than 85% of the population with more than 70 years. This is at the country level. And more than 75% of the same population have received the second dose. Our cohort of our first vaccinated cohorts, it's now on the second or third week of follow-up after the second dose. So we should be able to start seeing signals now. And the last one of the last slides, the idea here is just to show you why we think that we can provide really important data. So in spite of not being the country that has used more vaccines for COVID, the share of the population that's been vaccinated, it's one of the highest in the world, actually. And we have very, very detailed data of everyone that has received the vaccine, and also about the surveillance of COVID. We have a data set with more than 9 million observations regarding confirmed and ruled out cases. And also we have all the data sets coming from the National Health Insurance that covers 80% of the population. So those three data sets allow us to have a very good idea about what's going to be, the real world efficacy of the vaccine. So close for just last two slides. I want to show you how are we estimating efficacy, real world efficacy. And what we did was to design a cohort study. We exclude all those subjects that had COVID in the past, no matter when. And we included those that were part of national health insurance, which is most of the population. and we defined exposed and unexposed subjects according to vaccine exposure, of course. We are matching by age, sex, and location, and also by proxy for socioeconomical status using the type of health insurance they have within the public health insurance. And the outcomes are any infection, symptomatic disease, hospitalization, ICU admission, and death. So this is our Gantt. We started before the vaccination program was launched in early January. The follow-up started on February the 3rd. We are still following subjects, and we hope to be to continue following them for ever, maybe. We are now conducting our preliminary analysis, and we hope to be able to report our full results. Results on May. And then we are also working with different partners to nest additional studies using the platforms that we have in place. So, that's the most important part of the presentation. I would be happy to come back later with efficacy data. Just can tell you that you know, there are good signals after the first shot, and those get better after the second one. But we need more time for being sure that you know, there's always a risk for regression to the mean is always a risk. So we need more time to be sure that what we are seeing it's you know, solid. So this is the group that is working, mainly people from the Ministry of Health, our national immunization plan led by Cecilia Gonzalez. She's, you know, the one that elegantly has executed the program. Our data analysis group, our epidemiology department, our statistics department, many divisions working on this. Our public health institute, they are working on safety data, and also our authorities that are leading the efforts and also supporting our work. So that's all.
And thank you, Dr. Rafiu. Although it's still too early to judgment the effectiveness of the vaccines, but actually it's always great for us to see so smoothly large vaccination campaign happen in Chile. So so far we have already have the data from Brazil, Turkey, Indonesia, and Chile. Actually, this vaccine was first used in China. And so far, more than millions dose vaccine already used in China. So today, we also invite Dr. Zijian Feng from Chinese Center for Disease Control and Prevention to sharing the COVID-19 vaccination program in China, and also the surveillance data collected so far. Dr. Feng is the Deputy Director General of Chinese CDC, also the Vice Dean of Graduate School of China CDC, and currently he is in charge of EPI, emergency response, environmental health, and occupational health. I believe a lot of people are expecting Dr. Feng's presentation. Now I'm hand over to Dr. Feng, please.
谢谢孟总。
Thank you very much.
请原谅我用中文
I apologize that I have to speak in
各位
Chinese.
早上好，
Good
晚上好。
morning, ladies and gentlemen.
非
Good
常高兴
evening,
有这个机会
friends,
能够向
and
各位
it's my great
专
pleasure
家、各位同
to
仁
have
介
this
绍
opportunity to
中国的
introduce to you
新冠疫苗的接种情况，以
the
及
vaccination
我们的针对新冠疫苗开
of
展的
the
AEFI
COVID-19
的
vaccines in China,
监
as
测
well
的结
as
果。
the FIA surveillance results in China. My presentation falls into the following aspects. First of all, I would like to talk about the epidemiology of the COVID-19 in China, and then the vaccine development, and then I would like to talk about that in the future. What about the vaccination strategy and implementation results? And then I will share with you the AEFI surveillance results we got at the moment, and then some of the considerations for the next steps. About the current situation of COVID-19 after the outbreak in Wuhan, it experienced like 7 to 8 months in March. The spread of the virus was blocked in China, and now before the Chinese New Year, there was some small-scale outbreak of 25 cases, and those were usually, mostly caused by the imported cases to some local outbreak, like a dozen of them or hundreds of them. But for each outbreak, the duration was usually within 4 months. For weeks or within 1 month. So the local transmission was blocked. So the whole nation will recover to the zero case situation. On this slide, you will see those yellow or red colors. Those are the confirmed and the clinical diagnosed cases, imported cases, and quarantined at the borders. And the blue ones are those domestic cases caused by the imported cases. Now China is still in the transmission and blocking period of time.
中国采取的这样的一个传播阻断，就是我们严防死守，就是保持传播阻断的这个状态呢，它为我们下一步的那个这个实施推动这个疫苗接种，建立人群免疫呢，创造了一个非常好的一个环境，就接种的环境。我们可以相对比较从容的去推动疫苗的人群覆盖。那么在这种情况下呢，我们其实我们的接种的优先性的考虑呢，它有一个基本的前提，就是大体上在中国呢，各类人群、各个年龄层的人群，甚至包括各个职业的人群，它的暴露感染的风险其实是相似的，没有太大的差别。这跟有疾病持续传播的国家呢，它的形势面临的流行病学形势是不同的。但是呢，我们仍然要对这个接种的优先顺序呢，做出一些安排。那么目前呢，我们这个做出的主要的这个接种的安排呢是大概是这样。一会儿我要给大家介绍了。那么最终呢，我们的接种目标呢，现在是由于我们处在这样一个传播阻断的状态，我们可能不能像其他的有疾病传播的。持续传播的这个国家呢，能够观察到疫苗的它的对疾病的影响，那么也难以决定在这种情况下，就需要决定在中国未来接种的策略，等接种的目标达到什么样的水平的时候，我们可以这个开放我们的这个入境人员控制。和国内的这个防控措施可以做出重大的调整，这是给我们未来的决策呢带来一些困难。这是双重的，一方面我们可以在接种推动上可以相对从容一些，另一方面呢，就是我们需要做决定，就是将来我们的接种的人群的覆盖的目标或者人群免疫的类型的水平应该如何设定，这是我们的一个面临的一个挑战。
So those are the challenges we're facing with.
那么目前呢，那个中国呢，现在已经有这个多种这个疫苗在研发，就是14种疫苗呢正在研发的过程中，其中呢，有7种在目前已经完成或者正在进行在海外呢开展3期的临床试验。
third phase clinical trials, including for inactivated vaccines, including that from the Sinovac, and also we have one adenovirus 5-vector vaccines and 2 protein subunit vaccines. The Chinese government has approved 3 inactivated vaccines, including from the Beijing Institute, the Sinovac, and Wuhan Institute. So there are 3 inactivated vaccines approved with the conditions, and one more approved, which is an adenovirus vector vaccine. Recently, another CHU protein subunit vaccines has also been approved. So in total in China, we have 5 vaccines which have been put into the vaccination process. So those are the immunogenicity for the phase 2 trials. Those lab data can be found in a lot of published papers. I'm not going to the details. For the third phase trials, as it was already introduced by the speakers, results for Sinovac, I'm really grateful for the previous experts for contributing to the third phase trials in overseas, and with their cooperation, we got really important results. And for the inactivated vaccines, one is from the Beijing Institute, and they did the clinical trials overseas. That's the first one with 2 shots, and with 21 to 28 days of schedule. The overall efficacy is around 80%, 79% to be more specific. And for the inactivated vaccine from Wuhan Institute, 72% of the efficacy, and another one introduced already by Sinovac with different results. I believe that you have already heard the introduction. And another one from the adenovirus from a casino. The protection for 28 days, 85%, and 14 days, 68%. And for the protection to severe cases, over 90%. So it's quite similar to the result of a Sinovac, but there's just one injection. As for the current vaccination policies, it's already quite clear. At early stage, like in July last year, China has approved the emergency use of the 3 COVID inactivated vaccines according to the vaccine management law. So in certain group of the population, we started the vaccination, especially for those travelers and people working overseas and those working on borders. So we started the vaccination on those group of people for the first group. And starting from December last year, we started to launch the vaccination program and gradually expanded the coverage, mainly targeted at those people with higher risk, including those ACWs and those people on key positions working on the onset of the epidemic. And those who maintain basic operations of the society, and those working in the transportation sector, firefighters and police. and those people with plans working or studying abroad. As for the next step, it is quite possible that with the coverage of the first cohort of people, we're going to expand them to wider coverage. Those people with a higher risk of transmission, with more contact with people, working in more dense population, those people will be prioritized in the vaccination plan. And that is the policy for the first stage. And then we will actually open the vaccination to the other groups of people, including the elder people. In China, in terms of vaccination, there's one difference in our policy. So far, actually, we mainly cover the professional people. And in terms of the coverage on the people aged above 60, they are not in the priority group, because we consider that the reality about the COVID-19 transmission in China, and we also considered about that for the population aged above 60. So far, we don't have adequate data for clinical 3 trial. So we're very precautious in that. We hope that in the future steps, while we acquire more data about the efficacy on the elderly people, as well as people with chronic diseases, as well as the people aged under 18, then we can vaccinate those people and optimize our vaccination policy. These are some challenges for vaccination strategy in China. So there are very limited efficacy data on inactivated vaccines for the elderly, aged over 60, and people with underlying diseases, as well as the pregnant women and juveniles, etc. We need to get more data about efficacy and safety. And you can see that in China, it is very difficult to conduct clinical trials and VE studies that depend on COVID-19 disease outcomes, because there are so few COVID-19 cases in China. So we also look forward to acquire more data from the partner countries. We hope that we can have more observation-based studies and research with our partners in other countries, so that we can have better understandings about the efficacy and safety of our vaccine, so that we can further optimize the vaccination policies and strategies. And now in China, we actively prepare and implement the COVID-19 vaccination campaign in China. The Chinese government also formulated very detailed technical guidelines for vaccination, and we also provide trainings to almost 0.7 million vaccination professionals. And we also set up 27,000 vaccination sites for COVID-19 vaccination. At the same time, we have to deal with some possible acute AEFIs. So we also want to ensure the on-site emergency treatment for acute AEFIs. At the same time, in China, we also have the policy to provide free vaccination for all residents, 1.4 billion population, both vaccine and administration. At the same time, we also set up the electronic vaccination certificate, EVC system, so as to record the vaccination. And this is the COVID-19 vaccination progress in China. So far, 74.9 million doses administered, and 45.9 million persons at least one dose, and 28.9 million persons completed 2 doses. This is as of March the 19th, 2021. So in terms of the total number of population vaccinated, we're ranking number 2 in the world. And you can see that actually our company also donated a lot of vaccines to the other countries. So you can see that the population in China vaccinated are even less than some of the countries we're assisting. So we think that it also shows China's attitude. We also want to help the other countries. So this is China AEFI surveillance system. So far, the operation of the surveillance system is running pretty well. On the one hand, you can see that the healthcare workers as well as the relevant stakeholders will report the AEFIs, and the local CDCs will also have the investigations and have the initial diagnosis about the reported AEs, and there will be the causality assessment. So this is how this surveillance system operates. So far, we should see that this surveillance system is quite reliable. Besides AEFI surveillance system, and recently, according to WHO suggestions, we also strengthened the AEFI surveillance and response. So you can see that there's a strategy for COVID-19 vaccine safety surveillance in China, and we also selected some geographies to conduct causality studies, and everything has been going on smoothly. And now you can see we have actually 50.6 thousand AEFIs reported. The accrued rate is about 262 per million doses. So this 15.6 thousand AEFIs, 70% are common minor vaccine reactions, and the severe vaccine reactions is lower than 1 per million doses, and most were allergic reactions. So you can see that compared with national average AEFIs rates for all vaccines or flu vaccines in 2019, this is pretty low. You can see that the incidence of common minor reaction was lower, and the incidence of moderate and severe vaccine reactions was slightly higher comparing with 2019. And the incidence of severe vaccine reactions was lower than 2019. And you can see that comparing with flu vaccine, the result is the same, because flu vaccine are also widely vaccinated in China. And actually, this is the results of AEFI surveillance. From 3 companies, the orange bar represents Sinovac. So the moderate and severe vaccine reaction is just 30 per 1 million doses. For severe vaccine reaction, less than 1 per 1 million doses. And there are also coincidental events and anxiety-related reactions and pending. And actually, the investigation is still going on for the pending category. So this is the result of AEFI surveillance. The result shows that now for the 3 vaccinated, inactivated vaccines, they are very safe. And in the future, we are going to accelerate the vaccination. Since April, there will be accelerated vaccination. We will hit our target as soon as possible. We want to build up the herd immunity as soon as possible. So there's also some urgency we would like to do, which is the data acquisition. We want to cooperate with our international friends and partners so as to get more data for the efficacy and safety of the Chinese vaccines. So we also hope that you can also have the real-world data as soon as possible so that we can have better understandings about the efficacy as well as the safety of the vaccines produced in China, because this is very conducive for the future optimization of vaccination strategy in China and the other countries. For example, the efficacies on transmission and on different age subgroups, as well as the duration of the protection, when and whether we should enhance the protection, etc. So this data are urgently needed. So I do hope that we can have the relevant cooperation with the other countries. And this meeting, this webinar is a very good opportunity so that we can enhance our communication and cooperation with each other. And we can also enter into more substantial cooperation in the future. So this is my presentation. Thank you very much.
Thank you very much, Dr. Feng, for such